Well, how you doing, everybody? You are with us again at Cyberhuman Frontiers. It happens to be a nifty day today. It's the first day of a new month, and we have been doing quite a survey over the last number of episodes, a little bit of focus on the brain, a little bit of focus on technology, and as technology and biology become ever so closely entwined, then the difference between the two starts to blur so that you kind of don't know where one leaves off and where one picks up. And there are a lot of pieces to the puzzle of how that all goes together. Before we really get into it, let us just get our initial disclaimer out of the way right quick. And we'll read this as fast as we possibly can. The points of view in this video is not to, the points of view and purpose of this video is not to induce fear, violence, harassment, bullying, or in any way do anything thereof the kind, but rather to share and exchange thoughts with other like-minded individuals curious about the subject, encouraging conversation and awareness. The opinions expressed here belong solely to your hosts, myself and TMC, and are not a representation of the channel Cyberhuman Frontiers. And none of our videos claim to give medical, legal, political, or financial advice in any way. The content is created for your entertainment. The channel may from time to time contain certain copyrighted works of which we credit the providers and the producers of that content, and you'll see notes always below our shows that credit those fine individuals who do their wonderful work, and some very brilliant people. We refer you to the Copyright Act of 1976, in which allowance is made for fair use in such purposes as it provides, and that's also listed at the bottom of this video. I want to kind of get right into it. TMC, how are you today?
Andy, I'm doing good. I'm doing real good.
Okay, good. All right, so we're working to pull a lot of pieces together. Our shows build from one to the next, and we'd love to just review, and we do a little bit of that. In fact, we're going to go to a few basics tonight, but I want to just start off with having people just think a little bit about building things. Nature builds things, okay? It grows plants, it grows animals, it refines them, it eliminates some of them go extinct, some of them adapt, they mutate, they modify. How does nature build, and how do we build? So if I could have you imagine in your mind's eye a wild scene of a field, let's say a field with some woods or something, and you're going to see a scattering and a smattering of all sorts of diverse life of many different types. Some of them really grow, very vigorously. Others are sort of on the fringe. And, from one environment or one climate or one season to the next, any one of them might take more of the lead. It's kind of like it's just a complete cornucopia of life of all sorts. And when you look at how it's all combined out there in nature, it kind of looks a little bit chaotic because there's so much. It's a very, very varied environment. That's kind of the strength of nature too, because depending on changes in environmental conditions, there'll be some way for life to hold on, and something that was previously not thriving in a new environment, all of a sudden will be able to thrive, and it will carry on that process of life, whether it be photosynthesis or whether it be consuming plants and being part of that overall cycle of nature. When we build things, we tend to start simply, and we tend to be very orderly, very structured about the way that we build things. If you were to now, in your mind's eye, consider farming or the agribusiness at the very large scale level, you would see acres and acres, if not miles, of rows of a particular kind of crop, and they'll all be lined up They will be straight lines. Well, in many cases, maybe. And it's very regimented, orderly, and it's much more monodimensional. It doesn't have that totally chaotic natural effect that you'd see if you just took a walk out into a meadow somewhere and just looked at it and said, wow, what kind of an insect is this? What kind of a plant is that? So that's kind of the way we do things. And that's the way nature does things. I would like to suggest to you that the way that we are zeroing in on the code of life and the way that we're zeroing in on the integration of technology into the code of life is very orderly, and that it will have more of a tendency to be not a completely chaotic diversity like nature, but not totally monodimensional, but very, very limited spectrum oriented, because that's how we do things. We do things for a purpose. If engineers were put upon to try to build every possible variation in nature, they'd have a real hard time doing it, because the chemistry and biology of life is really pretty complicated. So we start small, start simple, and that's where we're making our inroads. And we're going to discuss a couple patents and some videos. We're going to start with some basics, and I think I'm just going to get right into it rather than talk on and on forever here. I'm assuming that many people here may know some of the basics about the building blocks of life, but this is part of the foundation of what it is that we're trying to work with. I'm going to put this right up and we'll stop it. It's not really very long. It's kind of short, but it's just a kind of a review of our, the molecules and the atoms and components and chemicals that make up pretty much everything that's alive. See if we can get this thing up here for you. TMC, any initial thoughts?
No, I'm looking up another patent that just came to mind, looking at therapeutic resonant frequencies, particularly with DNA and RNA, as you were speaking. So interesting. And that's all right. So I've got a few videos. They're all basic and they're all pretty short. So here we go. Here's one. This one's kind of musical, and they're very entertaining, and the animations are really good to help us understand what those component parts are. And now if you can imagine engineers in the background who are really getting into it, and they're saying, well, okay, exactly what kind of an atom is at this location on that protein? And so they get real deep into it. But here we go. Basics. Enjoy.
Go ahead and think for a moment about your very favorite food. What is it? Pizza, macaroni and cheese, chicken salad, sushi? Well, we all have different food preferences, but food is a source of large molecules that are needed for life called biomolecules. There are 4 major biomolecules that make up all of life, and this will be the focus of this.
There you go right there, and there they are right away. So there are molecules. The atoms make up molecules, and molecules in specific order make up either carbohydrates, which are our major fuel source, lipids, which tend to be structural and are energy storage, proteins, which are seriously structural, and then nucleic acids, which are the storage of the code, the order, the software, the instructions for building and assembling all this stuff. Let's let her go.
Video. Before we get into details about the 4 biomolecules, we need to talk about one very important vocabulary word, the word monomer. A monomer is a building block. If I had some large substance, the parts that make up the substance are called monomers.
Note, monomer is different from molecule. A monomer could be just one molecule, but generally speaking, monomers are groups of molecules. And so when we get into some of the detail about what we manipulate, what the engineers and the people in some of these research institutions are manipulating, they're looking at monomers, how they combine into groups of them, which guess what we'd call those? Polymers. Polymers, monomers. We go on here.
Just like building blocks. We're going to talk a lot about monomers today because we need to understand what the biomolecules are made of. And we need to understand biomolecules because they're building components of life. So let's introduce the 4 biomolecules now and talk a little bit about their functions. We'll start with carbohydrates, carbs. Well, carbs are something that you've probably heard a lot about when people are talking about diets. You know, they try to go low carb, or maybe they want a lot of carbs. Diets always come and go. Pasta and breads are examples of foods heavy.
I think you'll allow me to skip ahead because I just want you to grab onto the main categories here. I love the cartoons. Okay, carbohydrates. Okay, so we go a little further.
Monomer, again, remember, monomers are building blocks. The monomer for a carb is a monosaccharide. I know that's a big mouthful, but monosaccharides make up carbohydrates. Next one up is a diverse group known as lipids. Lipids are better known as fats, and they have 2 different types of building blocks. One type of building block is called a fatty acid, and the other type is called a glycerol. Now, examples of lipids include butter, oil, and cholesterol. Lipids, though, they have a lot of great functions. You might think, well, that's fat. How good can fat be? Well, it just depends when you put it in.
All right, so far we're on the saccharides, and saccharide, part of the root of that is associated with the word sugar, and sugars, complex sugars, simple sugars, saccharides, monosaccharides, polysaccharides, and they tend to be those carbon rings. They are generally pretty much rocket fuel for the cells. They are your high-test gasoline. They're not as much a structural component in the body. Now we're talking fats.
Into context. For example, you know those really adorable seals that you see on calendars? They have this fluffy white hair. They're actually called a harp seal. Well,
I love it. I love entertaining videos. I really do. So this one is from the Amoeba Sisters, and we've listed their channel down here. They are on YouTube, and we just want to thank the Amoeba Sisters for creating this little work. I do like it, and I in the interest of time and what we have to cover, I may skip ahead a little bit here. But that's not to say that you shouldn't watch the whole thing to your complete satisfaction along with the other ones.
Born with that keeps them warm, but over time they have to develop blubber. It's fat and it helps keep them warm. Lipids are great for insulating. Also, you might not think about fats as being related to energy, but fats are a great source of long-term energy. They can.
Okay, so I'll supplement what she's talking about here a little bit. Fats are good long-term energy storage. Gram for gram, they have more energy stored in them than carbohydrates, and it tends to be slower release, and it needs to be broken down a little bit more to get the energy out of them. But also, fats are part of the structural components in our bodies as well. Certain fats act as insulators for nerves, very important, because you don't want the electricity in your nerves leaking out. You want to get it to where it's got to get to from point A to point B, and they're also part of our cell membranes. And we're now in a very modern age where molecules are being designed that have, for example, with some of the medical technology that's being offered to us. I won't mention the name of what it is, but certain companies and certain formulations have been developed, which are incorporating some of these principles. In other words, the use of a fat capsule to deliver something that's inside it to a cell. And I'll let you fill in the blanks on what that is. It's kind of stuff that's all over the news and, all that business. So that's kind of what they work with. They do use fats as capsules.
Store energy for a long, long time. Say, for example, you wanted to swim the English Channel. That's like 21 miles of swimming.
Now, Amoeba sisters, forgive me, but I'm going to skip beyond the English Channel, and I'm going to go a little bit further ahead.
Hydrates. Remember, carbs are the fast source of energy. Well, you might not have enough energy storage unless you have some lipids on hand. Lipids also make up cell membranes, so they are very important for life because all living things are made of cells. Of course, an excessive amount of lipids could be
I'd like to kind of bring that idea to life at the macroscopic scale. If you got a pan of water and you put a oil droplet in there, you see that they kind of separate from each other. Now reverse it. Let's say that you made a bubble out of fat, okay, and it could be done. And inside that bubble, you confine something that's more water-based. Well, you know that fat and water don't mix very well. They tend to separate themselves, as you'd see from the droplet in the pan. Well, it's really a handy thing to use to encapsulate the more aqueous or water-based materials that are in a cell by encapsulating that inside of a fat cell or a fat-based cell membrane. And that's kind of why it is. It tends to keep the liquid that's inside the cell in, and it tends to keep the liquids that are outside the cell out. So the cell can be very specialized in what chemical balance it has internal to it. And I think it inherently makes sense if you think of that example. Let's let her go a little further. We're halfway through her discussion here.
Bad thing for your health. Remember, it's all about moderation. Okay, next, proteins. Now, when you hear about proteins, a lot of times you might think about power bars. They say they have a lot of protein in them and that they help with muscle building. Well, protein is great for muscle building. Examples of foods that are high in protein, sometimes you see these labels that say, this has 20 amino acids in this food. And really, they're just...
Forgive me, I did skip ahead a little bit.
Trying to say that it has protein, because proteins are made of amino acids, so that's just some fancy advertising for you. But in addition to being important for muscle development, protein is also very important for other functions, such as working in the immune system and acting as enzymes. Remember, enzymes are made of proteins, so proteins are important for the body. Now, when we start talking about genes...
So, something to keep in the back of one's mind is, you've heard the phrase, we are what we eat. And though whatever types of fats and carbohydrates we import into the body are what we're giving to it to use to make whatever structures it's got to make. So, quality of intake. Anyway, continuing on here. I don't want to get too much into the life sciences on the show.
Genes, the DNA genes, not the genes you wear.
We're going to back it up just a little bit here.
Enzymes. Remember, enzymes are made of proteins, so proteins are important for the body. Now, when we start talking about genes, the DNA genes, not the genes you wear, the DNA codes for proteins that are very important for structure and function in the body. The last biomolecule is known as a nucleic acid. Nucleic acids include DNA and RNA, which we'll get to when we get to genetics. They have a monomer called a nucleotide. That's going to be an easy one for you to remember because nucleotide sounds a lot like nucleic acid. And if considering DNA and RNA, both of these are involved in genetic information for the coding of your traits. They are found in a lot of your food because whenever you eat something that came from something once living, it can still contain the DNA. For example, when you eat a strawberry, you're actually consuming all the cells that make up that strawberry.
So that's something to think about when we get into the subject of genetically modified foods. It all kind of evolved in a balance over, either the process of creation some thousands of years ago or by evolution over millions of years. but there's a balance between what the other forms of life have built into them in terms of nucleic acid code and what we're importing once again into our own bodies, which will, to some extent, maybe little bits of that might get incorporated into us or maybe not. When we modify the original genetic code, or, the heirloom seeds, and make a special one, we actually really don't know. A lot of people think it's completely harmless, might even be good too, but we kind of don't know what the implications of that might be. But if you remember, the general thrust of our show here is we are now able to build basically the building blocks of life, and our laboratories are able to be at the point now of indeed building life forms that never existed before, and modifying the ones that we already have, such as us. So designing molecules and nano-sized nanoparticles and tiny devices, and even going further, building tiny structures that on demand can self-assemble into a larger structure once they're in a body, if they're given the proper input or the proper signal to do so, then we're getting into some really amazing stuff. It's all in the design. It's all in the code, the molecular code and the genetic instructions, which essentially is just a living form of software. Okay, genetic code is just some really complex and very ingenious software. But we're at the point now where we have the power to create life. So that just kind of got me thinking about that when she was talking about the nucleic acids.
And in the nucleus of all those strawberry cells is DNA. Plants and animals both have DNA. In fact, any type of life must contain nucleic acids to direct the cell's activities. So we just powered through introducing the 4 biomolecules by providing examples, exploring their monomers, and giving some general functions. One
last thing. I was just going to stop it, but the Amoeba sisters have one last thing.
Last very important part to mention is the structure of these biomolecules. Understanding the structure can help with predicting their properties and...
Okay, that's extremely important. She's right. Because some of what we can do, or not we as in me and TMC, but some of what can be done in laboratories is controlling the way that the same group of molecules or monomers are stacked, shall we say, or folded, or put into a conformation, which is just a fancy word for folding. And they'll have one function if they're folded one way, and they'll have another function if they unfold or fold a different way. And so when we have the ability to input something into the organism, which alters the folding and changes it to something else, we can thereby alter the function of it and make it either stimulate or inhibit the operation of what that molecule might do. And life does this too, by the way. Normal life processes will fold and unfold molecules when the time has come for it to do some sort of action for which nature designed it to do. So I think that pretty well covers this. We'll let it go another 10 seconds.
Easily being able to identify them. One thing I like to tell students to do is to write the 4 biomolecules in the same order that we went through: carbs, lipids, proteins, and nucleic acids.
Okay, there you go. So I am going to terminate this particular one right now. Thank you to the Amoeba sisters. Here's your rocket fuel, carbohydrates. Okay. Here is, and that's short-term energy. Here's long-term energy, and a very, very, very important structural component. Now, you've heard about bad fats and good fats. Well, if you're eating some sort of synthetic bad fat, and your body, you keep giving that to your body to have to make the envelopes that it makes all of your cells out of, then if you give it a fat that's not something that is akin to a normal life fat, then it might just make cell membranes that don't have as much structural integrity, or might be different somehow. And so, once again, eating the right foods. Protein, very structural component that gives rise to the proteins that are in, well, most important structures, such as your muscles, and complex proteins. They become other things? Enzymes are proteins that have the capability to act chemically on other molecules and make them do things. So enzymes are made of proteins. And then finally, the software, nucleic acid. So there you go. That was that. TMC, any wonderful and lovely thoughts from that little feature? Just basics, basics.
No, it's pretty good. I think it's good to keep driving the center point, if you will, of biology at this point. Biology, because that's where the real focus is. It's biology.
Right. So an intensive amount of research is being done on the code of life, and the code of life controls how we act, how the organism will act on any of these other molecules, or what it will build out of them.
Express is probably a good word.
Yep.
Whatever they will express, whatever the subcomponents will express.
So it's good to hit some basics. I'm going to move a little bit forward now. And we are now in the world in which nano-sized devices can be made that are literally at a very primordial level. They're like little robots. They can be connected with electronically or electromagnetically and controlled from the exterior, and they can act on cells, or they can act inside of cells, because they've been able to make some of these things that small. And so pretty cool stuff. It offers a wonderful frontier of targeted delivery of a therapeutic drug or chemical, to basically, target right to a cancer cell and wipe it out, things like that. The sky's the limit for what we can do. And we're kind of going to move toward the sky as we go through the show here. I got a nifty little cartoon. It's a cartoon. It's an animation, but to give you an idea of, I'm only going to just play a little tiny bit of this. I might not even play the audio. But this is what we're headed toward. In addition to merging technologically connected nano devices to the outside world with biomolecules and blurring the lines between where the artificial starts and where the living leaves off, we also have laboratories working on things like this. Now, this is a cartoon, and of course we have the link to this down in our show notes. This is a long video. I won't be able to play it, but this is from the Harry Messel International Science School, and it's from last year. And this is a little bit of it run, but I mainly want to just show you the little animation.
This is a picture of a blood vessel. This is like a red blood cell here. How big are these things? So a red blood cell is 6 microns. So 6.
Okay, these are red blood cells. These red goopy things here, 6 microns. Okay.
By 10 to the minus 6. So we're getting in the regime of nanometers. 6 microns is 6,000 nanometers. So we could make a nanorobot.
6 microns is 6. What did she say? 6,000 nanometers, right?
I think they call that an order of magnitude.
Yeah,
right?
yeah.
A micron versus a nano.
Yep.
Nanometer versus a micrometer would be an order of magnitude. I don't know. So the bottom line is that it's 1,000 nanometers is 1 micrometer.
Yes, correct. That would be correct. Yep. And this right here is 6 microns or 6 micrometers, if you can see that.
And that's,
So that's the symbol for micro, and that's why she's talking about 6 because these are about 6 microns in size. So anyway, just want to show you the
It does
stuff.
something to your cells. And in particular, we're interested in this. So this is what we'd think of when we talk to cardiovascular surgeons or people with heart disease. This might be an early stage, sort of beginning of heart disease.
What she's referring to here is the deposits on the inside of that capillary wall. It's a clinging of basically a kind of a sticky contaminant, which gradually over time narrows the artery. But we digress.
And we can't detect this now. And we ran around asking a bunch of surgeons, what would you do if you could imagine being inside the body, if you could shrink your...
I'll read this. If I could miniaturize myself in the body, I could detect early treatable damage in coronary arteries when you're 25 years old and thus avoid premature death or other problems. Yeah. And this apparently now from the context, this would be a coronary artery, and this would be the beginning of a plaque on the inside of that artery.
Down, make yourself really small, and go into the body. Would it actually change the information you got and change how you treat patients? And that was a really exciting thing to do, to go and talk to people who have real world problems and say, what do you want something that could be like a nanorobot to do? And we've spoken in particular to the surgeon, Professor Paul Bannon, who we...
So this is how things start off theoretically. It's an idea. It's what we wish we could do. And now we introduce the term nanorobot, nano-sized little machine.
Work with now. And he said, basically, if he could miniaturize himself in the body, he could detect these small changes that signal early heart disease, and that would totally change how we treated the disease. So way less surgery and way less poor outcomes. And so that's really exciting to somebody who just likes making stuff because it's cool, to say we can actually make something that might be useful to a huge amount of the population. So I think about 30% of people will suffer from some complications from heart disease at some point. So that's the why.
Well, this is an artist's conception of what we would have inside that coronary artery. These things would have some way of detecting the plaque or the thickening or a different type of molecule on the inside of a capillary. Capillary generally should be lined with a specific kind of cell. It's a form of endothelium, which is very smooth. And when you start getting stuff deposited on there, it's got a vastly different chemical signature, and it tends to bind to that endothelium like glue. So there we go. She's got her robots now. And that's mainly what I wanted to show you.
This is now the future you blood vessel. These are future you red blood cells. And these are future you DNA nanorobots that are going to go into your body, go around, find things that are bad, and fix them so you don't need to have surgery. So it sounds like a pretty far-fetched dream. But hopefully over the course of this lecture, I can try and convince you that it's not.
This lecture being a 55-minute lecture, which we're not going to go further with. But what looks like science fiction on your screen when you're looking at a blood vessel, a coronary artery and a heart, and something that looks like Clanky the robot, yeah, it's an artist's conception of it, but it starts off with ideas, and little targeting devices have already been built. So I thought you might be entertained by that briefly, and we're going to move forward. TMC, got anything you want to inject in here? I guess I look toward the future and how wonderful it is for the future of healthcare, and then I worry in the background as to whether it'll be something that really is available enough for enough people that need it, and how much it costs, and whether you can really get that. Whether you can go to your local drugstore and just get a bottle of this type of stuff if it was properly designed, and you just released a whole bunch of those little, cleaning jobbies into your body, and they'll go and clean out all that arterial plaque. That'd be great.
I think it'll end up being something that's a tabletop factory in a doctor's office, where they could take a blood sample or a cheek swab or something and synthesize whatever they need to synthesize to understand you, instead of the patient in the room next door, and then make a targeted therapy with synthetic biotechnology basically, in a tabletop, all in one, what we would visualize as like a good sized office printer. What you would put into an office that would have like 15 people in it or 20 people in it.
Yep.
And
now,
it's a machine like that.
What she was showing there was literally, Clanky the Tank Man robots. But in actuality, but people in laboratories have the ability to build or to design specific types of life forms that will be built to go after and just, eat that stuff. One possibility might be to design a particular kind of bacteria that just, its purpose in life is to float around happily through your capillaries until it finds a piece of plaque on the side of an artery. And then it says, I want to eat that. And it gets rid of it. It's not a stretch at all. It really isn't a stretch.
What else is not a stretch is when you look at the contraindications of an average, 70-year-old, the number of medications that they have, upwards of 10, 15 pills. And you got to worry about this interacting with that and that interacting with this. And if these interact, then it's going to impact this other medication. But if this medication wasn't in there, then it's not going to impact the other. You see what I'm saying? Where it becomes a chemistry project just to give somebody their medicine or alchemy. Take that notion and apply it to this nanotech stuff, though, is what I'm getting at.
We're entering into a whole new era medically. What it used to be was very broad spectrum, very wide spectrum, and it might be okay if you're taking vitamins, but if it's chemotherapy in which hopefully only 5% of the cells in your body are cancerous and they got to get rid of them, well, it's got to be, injected systemically. It goes everywhere and it can cause wreak a lot of havoc and debilitation on the normal, healthy parts of the body. So with an old person, that starts to become more important. The idea of targeting it right to where you got to get to and using little life forms is a good way to do it. Designing bacteria that will go after a particular type of cell that has a particular type of marker on it that is the signature of not one of the ones that you should have in you, let's say. All right. And, the immune system does this already. It's kind of, I don't know, I almost want to say they're kind of hacking the technology, the natural technology of the immune system now. And, we won't name anything specific, but there are laboratories who have designed some particular formulations very recently, which do, in a sense, they really do hack the natural technology of the immune system to alter its function and to kind of, give it a strong message. And, so it's got a lot of potential. I have more. This happens to be, but we can go off into reading too, if you'd like, TMC. We've got a lot of information to get to here. In fact, we may get to this in a while. I'll just read you the title of this. And once again, the link to this is down below in our show notes. Christopher Voigt talks about synthetic biology, programming living bacteria. You design that bacteria, you program it, you build it to have a certain purpose. And when you put that into the organism.
I think we should watch this before we read. If we're going to read about some nanotech and are you thinking the remote triggering or the self-assembling? Is that what you want to get into?
Yeah.
I think we should watch this first.
So and I think you got a really good point there. Indeed, there were a couple different ways to do these things. One is to zoom in from, I got to turn something off here. It's beeping at me. One way is to zoom in from the large size and wind up about at the size of a bacterium. Okay, that's one way to go. The other way is to design something from the ground up. Now, if I didn't say, and you were going to fill in the blank and it was a quiz question, then you would answer, okay, designing it from the ground up is starting with the code. That's starting at the nucleic acid level of what is going to get built what's going to get manufactured, and I got to shut this thing off. Okay, I did it. And telling cells what to become and what to grow into. And the cell, in this case, could be a bacterium. You could grow something like that. It wouldn't even have to be as big as a bacterium. It could be much smaller. You could grow something that was on the order of the size of a virus particle. And many of them only have a few thousand data entries in their RNA code and only have a very simple fat globule that they're encapsulated in. Some of them very simple. Relatively simple anyway. Anyway, yeah, let's go to this. And we're going to kind of start zeroing in on where it has been going and where it's at now and how far it can go. Okay. So we're talking synthetic biology. Here we go. And he's starting off macroscopic, as you can see. And let's see what he has to say. This is Christopher Voigt. Thank you, Christopher.
And hydrogen and so on. And it is really a very simple thing that we might be able to get. What we want to be able to do is actually fully unleash the potential of biology. And there are all sorts of things that biology can do that we don't know how to really access yet. So they can make chemicals that are impossibly sophisticated for organic chemistry, or build entire organs like the liver. It's a natural nanotechnology. So cells can build materials out of silicon or iron that are...
Yes, so you're connecting the dots. Cells do nanotechnology now already, but it's highly complex, and it's highly complex because the genetic code, the DNA, the instructions have so much information in them about exactly how to build that thing.
Are sophisticated and really have a level of accuracy that extends well beyond what we can do in the lab. And then finally, there are all sorts of functions that we'd like to be able to access. So at the bottom of the slide, I'm showing cells or bacteria that can associate with a plant in order to deliver fertilizer to that plant. And that requires real sophistication in the way that the bacterium is interacting with that host. And so the reason that we can't access all of these yet as products of biotechnology is really for two reasons. The first reason is that all of these functions require many genes, and all of the products that we currently get out of biology are maybe 2, 3, 4, 5 genes, something like that. Whereas these types...
Now, this is as of 2016. So enter now in the era of more powerful computers, supercomputers, quantum computers, and enter in the amount of, just the amount of hard work and how many people are focused on this. So you've got teams and teams of people, big institutions, working on cracking that code and working on, now we've already pretty well mapped out the human genome, but now working on manipulating that code by learning what every one of those, it's just, you imagine a really, really long number, really long, I don't know, a few billion digits long. And you're going to want to become a specialist in what does just this couple of hundred digits in that billion digit string mean? That's kind of what's going on. And people would never be able to do it by themselves, but with computers we can. And that's what's going on right now. And when we can do that, now we can start building stuff. And we can build interactive molecules that have other characteristics, which is what we kind of feature in Cyberhuman Frontiers, which is they can be controlled electromagnetically or by way of light or heat or acoustic waves. So he's going on here.
Types of products require control over hundreds of genes or even thousands. And then it's not just enough to be able to control those genes. You have to tell each gene when exactly to turn on, the timing, and also the location. So the particular environmental conditions where that particular gene has to turn on. And so this is really what we're trying to address in our work, where we wanted to create a language that allows a biotechnology person, somebody that's doing genetic engineering, to go into a cell that they're trying to build and tell exactly what genes to turn on at what times as part of building these products. So now if you're not used to thinking about cells as doing computation, they're actually doing computation all of the time. They're thinking and processing about their environment. They're figuring out where they are. They're figuring out what genes need to be turned on to survive. And the way that it's doing this computation is that it uses a really large, sophisticated regulatory network. And a regulatory network is just where you have proteins, you have DNA and RNA that are all interacting with each other. And within these interactions, you then get computation that arises.
TMC, you're able to hear me right now?
I hear you.
Just changed microphones.
Sound great.
So he's speaking about something simple. He says a simple regulatory network. Now, that's a whole lot more complex than just looking at, the four major cell types of life. And what we're really seeing here is, and I'd almost would like to zero in on it a little bit more. Before I do that, let me see if I can pull it at higher resolution, which I think it can do. Yep. And you're seeing logic gates here, okay? This is being expressed in terms of logic gates, which are computer gates. They're the stuff that's used, AND switches, OR switches, NORs, NAND gates. That's the language of computer technology. So when you combine together, this, for example, is a logic gate, and this is a logic gate as well right here. And using that type of terminology and combining that with the code that's involved, the DNA programming. So now you've got the software and the hardware. And what we're seeing is a kind of block diagram of what we're working toward is a block diagram in almost electrical engineering terms of life. That's what we got here. And now, if you can imagine, let me zero in again. And this is what's going on. We're going to talk about some of this stuff in tonight's discussion. If you can imagine one of these things, let's say this is just a normally functioning representation of some life process going on. Okay. Now, if you can imagine that you go in and you add an additional input to one of these things. So you've made it light sensitive, or you've made it sensitive to a particular vibration or a particular radio frequency, so that now you cause, let's say, this particular gate to ignore one of its inputs based on whether it's being blocked by a certain specific frequency. Or imagine on the output side of this. Now, this is the inputs, 2 inputs and 1 output. Imagine on the output side of it, when this thing turns on, that imagine if it excites a photoluminescent molecule. So you get an additional output out of that particular molecule, okay, which is it functioning in this case as a logic gate. All right. This being an AND gate, by the way, in case anyone was interested. Logic gates. So this is how we start. And let me also open it up so you can see what he's got at the bottom of this thing, too. It's all very interesting.
So what I'm showing here is one of the first regulatory networks to be figured out. And really, it's encoding a very simple decision that a virus makes when it enters a bacterium. So it basically has to decide, am I going to immediately kill this bacterium and escape into the environment? Or am I going to hide out in its genome to survive for a longer time? So this yes/no decision gets encoded by these different interactions. And so if you look...
We want to stop you right there and just say, always be connecting dots in the background. He's talking about the mechanism by which a virus decides what to do when it infects a bacteria. Well, viruses can infect human cells as well. And so that virus has got some kind of built-in intelligence at a very low level, if you will, that allows it to know whether to hide out or whether to become highly active and do its virus thing, because, that's what they do. They multiply themselves, and eventually they usually wind up killing off the cell that they use to multiply themselves. So there's intelligence. Even in viruses, there's intelligence. And so keep connecting dots.
Look at this plot. You'll see that there are circles, and these are proteins, and there are lines, and these are interactions, and the horizontal lines are the DNA that they bind to. And all together, they work together to create this switch. So at first, in the 70s and 80s, as this was being discovered, people started to use some language like switches and
TMC, let your imagination run. Everybody else too. You know what I'm saying? Think of it. Think of that merger between technology and life. Here it is. You're seeing a really good picture of it at a simple level, right?
Yeah,
Think of adding sensors. Think of adding more inputs.
This reminds me of the first Batman movie when they were breaking. Seriously, listen. When they were breaking down on the news report how the system worked, where the Joker put one chemical in this product and a different kind of chemical in another type of product, and you had to use the right combination of products in order to have this death basically befall you. And so when this guy's been talking about basically using biology as a circuit, as a logic-based circuit or a logic-based form of reasoning or computation. And that's what it reminded me of. It really reminded me of something that's binary. It's either off or it's on. If it's off, then it does this. If it's on, then it does this. And then you have derivatives out of that, combinations of off and on, certain orders of off and on, time in between, all of those things. And then you start like trying to apply what we understand as computer language or computer talk or computer concepts.
Yep.
And try to translate that over into biology. And most people can't do it.
But and that's what's going on. TMC, that's exactly what's going on now.
I have a hard time doing it, man. And I think that the reason why it's hard is because it's difficult to visualize the biology piece to be able to make that translation. And that's why I find value in listening to the last couple of shows that you've put together, man, is because this biology piece is really, really, really important. You could say all day long, hey, the technology's here, Internet of Things, and, we can see the ushering in of AI and computers and running everything. Automation is the term I'm looking for, up to and including putting people on the Internet. And most individuals hear that, particularly the last piece where humans are going to be on the Internet. And then they just, nope, not having it, don't understand, does not compute. How is that possible? I'm not taking a chip, this, that, and the other. And it's like, well, it's way more sophisticated than that. If you can decode the entire biological organism that is the human and map it out,
there's some pretty logical stuff going on. Dude, yeah. There's actually some really logical stuff going on in there. It's just not apparent when you go and look at a fully formed piece of life, like a hummingbird hovering over a flower. You say, wow. I mean, that can't just be a bunch of logic gates, right? That can't be just simple logic. But if you go...
It's not. The piece that is not being discussed here that really is a gap at this point, and I think it's been overcome, but it hasn't been necessarily deployed and saturated, is this idea of probability. So you can have the logic circuits deployed all you want. You can have all the hardware, yes, no, switch left, switch right, switch up, switch down, whatever, right? However you want to visualize that.
Yep. I love where you're going with this. I love where you're going with this.
Probability, which ultimately from a purist point of view or philosophical point of view, is ultimately dancing around this idea of free will, but it's beyond or even it's more than just free will. Probability is like, am I going to slip off the curb? Is my foot going to slip off the brake while I'm in my car and cause an accident? You see what I'm saying? Probability, the likelihood of something happening or something not happening, and then that rippling out. If this happens, then that creates a ripple effect out into space. If it doesn't happen, that also creates an effect or doesn't create an effect, which in turn has an effect further out in the ripple, whether it's there or not. And so, anyway, I'm digressing.
No, you're not really.
This idea of probability is huge, And when we look at introducing genetic code into the environment that is man-made, and understanding that things can be dormant, things can be active, things can be assembled or disassembled, they can be cut apart, they can be misformed, malformed, reformed, remade.
Yep.
Right.
And life makes mistakes too, by the way. Yeah. Life screws up sometimes, right?
For the better or the worse, right? That's mutation or evolution for you. Evolution through mutation, which is just an expression that is different than what it came from. Simply put, I mean, it's probably more eloquently stated elsewhere, but that's really all a mutation is.
But then there's the interactive part. This Earth and all of creation, if there is, and in my view, there probably is life elsewhere. If you got one life form, one single isolated one, and at its most rudimentary level, it's functioning very logically. It's got some version, a much larger version of all of this complex interconnected logic circuitry in it, right? Now, there's another life form, okay? And the other one has got its own logic. But when those two encounter each other, then there's an interactivity between them. Now, think of what happens when you look out at that meadow that has this total cornucopia of all these different life forms, each with their own logic programs, albeit very complex logic programs, and then they interact. That just blows my mind. Because look at all the possibilities. Look at what can happen. And that's not even getting into, a merging between a couple of slightly sister species and coming up with another version. That's not even that. Anyway, I find this very interesting. Just the idea that these guys, and this is the speaker here is Chris Voigt. The link is down there. That these guys have been able to come up with a graphical representation such as this. So let's see what else he has to say here. In fact, I might back it up 10 seconds.
Logic that they're proteins, and there are lines, and these are interactions, and the horizontal lines are the DNA that they bind to. And all together, they work together to create this switch. So at first, in the 70s and 80s, as this was being discovered, people started to use some language like switches and logic and things like that. And then in the 90s, as these networks got more and more sophisticated, you started to see some of the language of electrical engineering being used in describing these types of networks. So if you look carefully, you can see that there are going to be some cases. So for example, if you look up here, where you see these little gates. And so these are symbols out of electrical engineering that describe a logic operation that's being done. So those red arrows are the signals that are going into that logic, they get processed, and then there's an output. And if you look at the natural regulatory networks that are in bacteria, they get huge. So what I'm showing here is this mothball is a interaction network for the bacterium E. coli, where all of the red circles are proteins and the blue lines are interactions. And it's all of these proteins and their interactions and the way that they interact with DNA that is allowing that bacterium to compute on its environment. And so somehow the cell is able to get hundreds of regulators to work together. Now, for about the last 10 or 15 years, a variety of researchers have started to create what are known as synthetic circuits. And this is where there's a circuit function that they may want to produce, like an oscillator, and then they figure out how to get the right regulatory proteins to interact with each other in order to create that oscillation that they want. And so, for example, I'm showing at the bottom of this slide where a few of these regulatory proteins are wired together to create an oscillator. The green little images are bacteria that are oscillating, and you can see in the graph the quantification of those oscillations. And so a lot of these types of circuits have been built that are oscillating.
And we know when he uses the term oscillator, we know that that just means something that has a cyclic nature. It goes on, it goes off, it goes up, it goes down. It goes from one state to another in a perpetual way. If you leave it alone, it'll just keep going, up, down, unless there's some other force that interacts. That's all he means by that.
Man, that'd make a nice little lobotomy right there. Seriously, you get to look at what they're talking about here. They're talking about fluorescent bacteria, but I mean,
right.
you have biofluorescence, and we went over optogenetics quite a bit, and this is completely hypothetical, and so just go with me on this fantasy for a moment here. But like, you didn't have to cut anything out. You could just offer genetic information into the body, have it strategically deployed into the brain based on what receptor sites you're targeting, and then build in bioluminescence and optogenetic responsive ion channels that are kind of in the same neighborhood, and then get these things like this guy's talking about to get them to oscillate at a particular
maybe a rate.
Hertz. Yeah, you put it down at like 7 hertz or 6 hertz or something, just above sleep, and you would get like this very tranquil, low energy, non-halfway sort of functioning kind of state of mind all the time. Like that state of mind when you wake up after you've just, like, you've been up all day, and then you go to sleep and you wake up 2 hours later for something, and you're like super groggy, and it take like if you had to like get up at that point and function, it would take you 30 or 45 minutes of just what the hell is going on right now to get to a point where you could like really, really function. I'm talking that kind of tired. Put your brain, think about where your brain's at when you're that tired, and what the brainwave oscillations would be. So if you could synthetically recreate that in the brain just by offering gene therapy, I mean, you could domesticate He-Man with some shit like that.
Well, couple that together with what we might hopefully get to. We're halfway through our time here now, but when we talk about the new IP numbering system, the predecessor was IPv4. Okay, yeah. Buzzwords, right? Okay. Well, IPv4, they started to see that they were going to reach a limit of how many digits they were going to have. So they came up with a new one, which is called IPv6, which now offers enough IP numbers, so that you can literally have an IP number reserved to just about every type of neuron that you would have in your mind. Okay. And if each one of those could be addressed individually, then that is basically the addressing scheme, the accessing and communication scheme for every single neuron in a person's brain. So now, if you are monitoring or interacting with those, you can do a whole lot in terms of mental telepathy and, or an electronic version of mental telepathy, and a whole lot of other things too. So, yeah, I mean,
Indy. I wasn't gonna say anything, but I gotta say something. If they had complete neuronal mapping down to every last cell, like you're saying, hypothetically, they're able to do that. It's way beyond synthetic telepathy. It's like controlling. You'd be able to very, very specifically control somebody's body temperature. You'd be able to very, very specifically control their respiration, their heart rate. You'd be able to selectively dilate their pupil. You would have functionality like you would operating or driving a vehicle, or even more so beyond that. It would be like
far beyond
stepping into a robot suit and it being a synthetic version of all of your 5 senses and moving around and functionality and motor function and all that.
Yep, the extensibility of it, the virtualization that is as real as anything could ever be. It could be so real and so extended that it's science fiction.
If they had every single cell, they would be able to potentially selectively fire or selectively activate
or inhibit
any of those cells in any combination that they wanted. So theoretically, artificial sensation, a completely artificial experience would be possible at that point. You wouldn't even need to move. All they would need to do is just offer stimulation protocols, and you would have a complete total matrix experience. With the
again,
scenario you gave. Yeah.
again, you have to ask yourself at that point, what is that physiological body really needed for? You know, what do you really need it for? To do physical work?
Yeah, to do some function, some task. That's what the body's at that point would be for.
Yeah.
You know, even if the task is only to procreate or to engage in any type of pleasure-seeking activity.
Well, one of the frontiers is the intelligence of the human brain, because it's got trillions and trillions of neurons, and it still surpasses what we can do with any computer that we have so far. Hint, so far. But once one has reached the point that one is completely interconnected with something like that, then that person's mind can become part of a collective neural network of linked together networked brains that might be the most efficient way to build the next computers, would be to just link a whole bunch of brains together and have them all under some sort of overall governing administrative control.
Well, yeah, I mean, that's really kind of why DARPA set the whole internet up in the first place. But, you know, a lot of people won't believe that. So
it seems so far-fetched, but I mean, you know, we just saw a diagram of the block diagram using electrical engineering terminology of a simple life form, a virus. All you got to do is scale it up. That's all you have to do. Just scale up, go up to a bacteria, go up to a eukaryotic cell, which are the cells that are in humans for the most part. They're more advanced cells. And then go up to specific ones. And once again, we keep coming back to neurons because those are the ones that kind of, you know, they connect and control so many of the other types of cells in our bodies. And neurons...
Well, the neurons are most important because they govern behavior and action.
They also hold memory. They hold knowledge.
I don't know. I personally, I think that's debatable.
Well,
they can. I currently subscribe to this idea that memories are really just accessing a particular state in the 0 point or the quantum field.
Okay, yeah.
Where it's a particular, you know, what you call it, overlaying of frequencies or whatever you want to call it. I don't even know how to like verbalize what I'm seeing in my head, but
we have, do you remember that really cool document that we have from months ago that we never got to the end of?
Yeah.
It goes in that direction.
Yeah.
And I have I wage no fight with you about that possibility. None.
Yeah. And well, the reason why I say it, and I listened, I think, to Mike Adams talk about it, and it was very compelling, but one of the citing examples that was provided, if it was Mike Adams, it may have been somebody else's analysis. But when people get an organ transplant, right, like you get a liver transplant or heart transplant, and all of a sudden you have memories that are from the donor life, the donor's
How'd that happen?
life.
That's right.
Have you heard those stories? So
yes,
how do you explain
I have.
that? If it's not a brain transplant, how do you explain that? And really what it is, it taps into the whole idea that your genetics, as you go forward in time, your genetics are changing based on your life experiences. And
Correct.
So it's not just what you eat. It's how you feel. It's what you experience. It's your emotional state. It's everything that has an epigenetic influence on the overall genetic structure that then becomes this hardware, software, kind of a firmware, sort of a memory chip that governs, you, if you will. But that then becomes, your offspring. So all of your experiences and all of your memories and all of your feelings and all of your emotions and all of the everything that have made your genetics what they are up to the point of conception is then transferred.
Yep.
As a boot, like a base layer, like a BIOS of available software code that your offspring can then access, or that is the software code that your offspring accesses as they go through their life, experiencing their trials, their tribulations, their successes, their happiness, their failures, everything. that then in turn taps into that genetic code to respond and to adapt and to I guess express as that your offspring in this case goes through their life. And so when you start changing this stuff, or when you start manipulating this stuff, it's way more than
So I guess one question would be, when that organ transplant happens, is there some additional bit of encoded character of the previous donor that is at a physical level, or is there something more at an extended level that you were talking about?
I think it's extended. And I think what happens is if you get a blood donor match and you don't get organ rejection, ultimately over a period of time, that organ is going to assimilate into your body. It's going to reproduce. The genetics are going to fuse together. You'll have enough time where cell replication will occur.
They'll merge.
And it'll start. Yeah. So the quantum entanglement, if you will begin to proliferate throughout, and it gives rise to that notion that you and I were talking about, where can a quantum ripple create a quantum effect somewhere else that has nothing to do with whatever change was made to ultimately create a quantum ripple. And then what does a quantum ripple do when it interacts with a different quantum ripple, and you can extrapolate that out over and over and over again. But
there are people who have said some very thought-provoking things. One of them was that all points in space are all connected, and all points in time are happening at the same time. Now, that is deep, deep, deep stuff, but that's going all the way to the edge of stretching our imaginations.
Yeah. Well, going back to the memory thing with the organ, I think once enough blending has occurred physically, I think that it's almost like a radio antenna. The receiver just kind of opens up over to that other frequency where it's kind of been hybridized on to some extent.
Yep.
And it's not, you could argue it's stuck in it's in the genetics and it's hard coded in there and or you could argue the other way, where the genetics becomes a unique antenna, basically a fractal antenna, but very, very unique, that then is able to tune to that particular experience.
Yep.
Which means that every experience technically has a place in the quantum field, which then takes you back to this notion of time.
Possibility as well. About
probability, also true. Damn.
About the ability for a person to change their reality, to change their future. Not going to alter one's past, but if one subscribes to the notion that all points in time all happen simultaneously. We happen to live in a terrestrial world. It's very concrete, and time is very linear. That's just the way we're built. That may not be the way it really all is. It's just the way we live through it. It's the way we experience through it. So anyway, back to Christopher Voigt. He's speaking in very concrete terms. And, this is what we do. I mean, we start off with simple, concrete things and then we analyze this. These are just amazing scientists working on this stuff. I think he does a really decent job combining together engineering technology and relating it to life forms. I want to go on with him a little bit more. Are you good with that idea?
Yeah, that's just fine. We're gonna have to share again, but that's just fine.
All right. Yep. Here we go. And back it up maybe 10 seconds. And absolutely fascinating. So let's back up to about there.
A few of these regulatory proteins are wired together to create an oscillator. The green little images are bacteria that are oscillating, and you can see in the graph the quantification of those oscillations. And so a lot of these types of circuits have been built that are oscillators or pulse generators or logic functions. Just about anything that you see in electrical engineering, it's been reproduced. But there's been a challenge. And that is that all of these circuits have been limited to 2, 3, 4 regulatory proteins. And we know that there's this possibility where the bacteria has hundreds of interacting regulatory proteins, but we haven't been able to bridge that gap to go from simple circuits to the level of sophistication that cells naturally have, which is what's ultimately going to be required to control.
I'm just going to stop it right there. Note the date. He said we haven't been able to. That was in 2016. Well, you can bet your bottom dollar that we're a lot further along that road now. What did I do? Oh, no. Where was I? Oh, there we are. Okay.
Oscillations, generators, or logic functions. But there's been a challenge, regulatory proteins. And we know that there's this possibility where the bacteria has hundreds of interacting regulatory proteins, but we haven't been able to bridge that gap to go from simple circuits to the level of sophistication that cells naturally have, which is what's ultimately going to be required to control them. And on top of that, even building one of these simple circuits requires years of effort and tends to be a very high-profile type thing to do. And so we wanted to both try to build larger circuits, but then also make it faster. And so first, what are the things that are stopping us from making this transition from a few simple regulatory proteins to getting it across the entire network to make things that are at the scale of genomes? So first, one of the biggest problems is with design. And this is where there simply haven't been enough well-characterized regulatory proteins where we understand how they behave at such a quantitative level that we can predict how they're going to work together as we combine them. There's also a problem where software doesn't really exist to help you with that process. And so researchers are forced to manually.
That was then. This is now. That was 5 years ago. This is now.
Put pieces of DNA together in a word processor to try to figure out how to build one of these circuits. And then finally, there's also a lot of toxicity that can arise, whereas you put these pieces into DNA.
But I think I got to bring back George Church talking about how they took, I think it was a book, a small book, and they wrote in DNA base pair code the whole book, right? So
they took an HTML of his book on genetics. I forgot what the name of the book was. I'm sure he's published more than one. Yep. But and they also encoded the Java. So it was a soft copy. It was a digital version of his book that had Java behind it to run the display. And all of that was encoded and then went through an experiment, which I guess was the encoding into something like yeast. And then they were able to sequence the genetics back out. Once the yeast had procreated, they reproduced and multiplied.
Right.
And they were able to extract the book again and the HTML code.
That's a really inexpensive way to publish multiple copies of a book, isn't it? Instead of Gutenberg.
It's a real inexpensive way to barcode the entire human population, isn't it?
It's very compact. It's very, very cheap. I mean, developing the technology to do this is rather expensive and requires a lot of brain work. But once you get that done, it's very compact, it's very inexpensive, and it's very, very, very scalable. It's
scalable, it's modular, and it becomes universal. This is a common language of life on this planet.
Yep.
Yeah, this is like they get this right. It's the Holy Grail. This is the Holy Grail they're chasing after.
This is a big, big takeaway also. There are many commonalities that us life forms share. Many, many repetitive building blocks that occur over and over again. And some that generally are not shared between species, but that we have the capability to import them into other species now. But there's a lot of commonality. Okay, he goes on.
Can start to hurt them. The second problem is just the physical construction. So building a big piece of DNA is actually quite challenging. And putting all of those pieces together without creating errors has been a big problem. But there have been a lot of technology recently.
So you remember last week, was it last week or the week before? Literally at high school or maybe college level, there was a a group of students working on building a plasmid. And a plasmid only had a couple of thousand pieces of genetic code in it. So, and it had certain segments, certain parts of it that are characteristically the beginning and the middle and the end, and certain active parts of it, and being able to design such a thing as that with a couple thousand base pairs of code. So that's happening now in schools for students to learn.
Recently that helps us address this. And then finally, going in and debugging one of these circuits is a challenge. We're putting them into living cells that change based on their environment and what stage of growth they are. It's often difficult to see how the circuit is performing, and you usually have to look at dynamic measurements. So you can't just take a picture of the cells and from that one picture know that the circuit's working. You have to follow it over time, and that can sometimes be a real challenge. So throughout this talk, I'm going to describe how we've gone in and addressed a number of these different challenges. And so the paradigm that we decided to do was to really go after creating a software program.
Okay, what we have here is a computer. We have a logic diagram of the genetic circuit. And then when we have that circuit, we look at what particular sequence of DNA is it going to require to build that circuit. And it's a little too small to see here, but what you would see is nucleic acid base pairs, either C, A, T, or G. It's a 4-digit code that is the code of all life on this planet, including, if you're talking about RNA, there's a 5th one called uracil. And then taking that code and then getting that into this little thingy, which is a bacterium. Okay, so computer, block diagram, the actual digits that it takes, and then get it into a life form. There you go.
That would allow a user to go in, and in the same way that they're programming a computer, they could program a cell. So the first step of this is that a user would go in and write a textual program that then the software compiles into a circuit diagram that's made up of individual gates that are connected together in order to create the circuit function that's desired. Then the software creates the DNA sequence associated with that circuit function, which then can be sent off and synthesized by a company, sent back to you. You put it into a cell, and then the program that was written in the computer gets run in the bacterium.
Okay, we're doing that now. Just in case, and this isn't theoretical, this is being done now. And it's pretty advanced.
And so there are 2 stages to get this to work. The first, of course, is just getting all of the software, and that was relatively straightforward. One of the most challenging things were getting the gates, or the fundamental units of computation, that work so robustly that you can put them together in different configurations to allow a user to create any program that they wanted. So if you're not used to thinking about how cells do computation, I have a little animation here that shows how we do it. So if you have a sensor, this is, as we define it, a piece of DNA that allows the cell to respond to a signal and then control the expression of a gene. And the way that this works is that in the...
So connecting some dots, pieces of DNA that can function as sensors, right, TMC? And making specific modifications to those pieces of DNA, which we do have the capability to do, to make that sensor sense whatever the most optimal type of input might be, whether it be acoustic, electromagnetic, or whatever it might be, right? So we have the capability to do that now. And let's see, he goes on. I don't want to interrupt his example. He's doing a great job explaining this, better than I could. Thank you, Chris.
Sensor, there's a piece of DNA that allows the cell to respond to a signal and then control the expression of a gene. And the way that this works is that in the sensor, there's a piece of DNA that encodes a gene that makes a protein that then can bind to the signal. So in this case, it's a small molecule shown in red. And when that protein binds to the molecule, it can then bind to DNA at a promoter. And this causes a flux of RNA polymerase that then turns on the gene that this is connected to.
Okay, did you get all that? So, signal comes in, it binds, and when it binds, it makes this thing turn on and sends a signal out to synthesize something that otherwise would not happen if the binding signal had not gotten connected to that piece of nucleic acid.
And so if you have a presence of the molecule, this causes a high flux of RNA polymerase. And in the absence of that molecule, there's a slow flux of RNA polymerase. And so in defining a sensor, we can then black box this and just think about the different levels of signal that's present and the different fluxes that this then causes. We can then define a circuit similarly, except that in a circuit, we have both the input and the output as RNA polymerase fluxes. And so in this case, we have a circuit that takes a high flux of RNA polymerase as the input, and then converts this to a low flux of RNA polymerase as the output. And so this acts as an inverter, and you can see the response function.
So do you draw any parallels to being able to get something to add functionality to one's existing brain neurons, for example? And you now have a way to not only see what that activity is, but to modulate the rate of the activity based on what you've done inside that black box to alter to either inhibit or to stimulate the response to the signal. That's in addition to just getting the signal. You know what I mean?
Yeah, but this, beyond just a neuron or a brain cell, this technology basically it's anything. So you're talking about gene therapy and organ regeneration, or regrowing a limb after an amputation.
What about instructing stem cells exactly where to go and what to build?
That kind of stuff, dude. That's where this kind of technology will go. Right now, he's giving you an example of something that they're going to do in a lab to measure flow rates. And you think of the word flow, you think water or liquid, but it's not. It's really just an expression of movement. Where they're trying to understand the basic concepts of how to translate electrical engineering into biology. Where this is ultimately going to go is radically advanced therapies.
Yeah.
And or radically advanced
absolutely amazing therapies.
Yeah.
We've just seen an example which he talked about of programming a bacterium. Now substitute that is one of your stem cells. Your stem cells can become anything. They are so if you can program a bacteria, you can program a stem cell. You can program that cell where to go and what to do. You can say, okay, this lady's pancreas is burnt out. She's got diabetes. We need to grow pancreatic cells. We are going to bring in some code and get it to her stem cells. And then those stem cells will eventually find their way to the right location, just like they did when we were little babies and we weren't fully differentiated yet. And all of those different cell types migrated and differentiated all over the body to become the specific structures that we have. It's the same idea. It happened to all of us when we grew from fetuses into bigger humans. Same thing.
And what's really key about this design is that because both the inputs and the outputs are the same, they're both RNA polymerase fluxes, it becomes very easy to connect them to sensors and to each other. And so, for example, you can have a sensor that has a signal that's an input and an RNA polymerase flux as an output. And this can be connected to a circuit that has both the input and the output as a flux. This can then be connected to another circuit. And this can be done ad nauseum until you're sick of it. And then the last step.
I'll go further, though. Now, right down at the bottom here, it's the thing is in my way, but if you pictured another row of circuits and you pictured a branch connecting two of these things so that you've got two input circuits connecting to one output circuit. Now you have the ability to create a logic gate. It would be an AND gate, whereby both of these two would need to have sent an output in order for the final one to deliver its final output, or it could be an OR gate. Either one or the other could cause this thing to fire. Or it could be a NOR gate, in which both of these have to be inhibited for you to continue to get the output out of this one. So that would be putting it into, once again, into, computer logic gate terminology.
Is you take an RNA polymerase flux as the input and convert this into a cellular response. And the device that does that is called an actuator. And so to figure this out, we turn to some theory from electrical engineering and something.
I hadn't looked at this part of the video. Now he's going to say what I just said, but he's going to say it very well.
Okay.
That had been recognized in that field is.
Say again, TMC?
Yeah, I really just want to emphasize that this part is really, really, really important. And it's math, and it doesn't make sense at first, and it's fine. But this is basic computer logic stuff.
It is. It is.
And this is what they're cross-applying over into biology. If this, then that. If this, then that.
Exactly.
If then. That's all it is. And it's logic, and it's controllable, and it's reproducible, and it's exactly the way software functions on a computer, and how logic and computation is done on a computer. They're doing it with biology. So
it's very important. Hide out in its genome.
Sorry. Hold on.
I'm done ranting.
This is a virus. This is the logic flow in a simple virus. So he's showing us how they got there. That's what it is. It's just, yeah. Yep.
To some theory from electrical engineering. And something that had been recognized in that field is that some logic functions are what are known as Boolean complete. And what this means is that anything that you can imagine on the computer can be broken down into these simple Boolean complete logic gates without any additional computational functions. It is one of the basic principles that allows digital computing. And so one of these functions is what's known as a NOR gate. And a NOR gate is a 2-input, 1-output logic function. So here I'm showing the electrical engineering diagram for a 2-input, 1-output NOR function. And then this is the logic function that this produces. So when both of the input signals are off, the output is on. And if either is on, where they're both on, then the output is off. So believe it or not, that simple function is all that's required to build anything that you see in the computer. So now that's sometimes hard to wrap your head around. So I like to use the example of the Apollo 11 missions to the moon. These were very basic computers at the time. And believe it or not, they were entirely based on interconnected NOR gates. So it took 5,600 NOR gates in order to create the guidance systems that took those astronauts to the moon. And so if we had 5,600 NOR gates that we could put into a bacterium, we could reproduce the Apollo guidance systems within a cell.
Let that sink in for a second. How about that, huh? If anybody is as old as I am and they remember how large and primitive computers were 30 years ago, and now what we have, this will move forward very, very nicely. And we're going to have very, very complex and very, very tiny, sophisticated logic arrays that can be inside of a cell or a bacterium, literally. Complex arrays of computing gates that we will be building. We will, well, not us, but these guys that know how to do this stuff will be building. Think of what you can do with that.
So the trick then becomes, how do we create a logic function, that's this NOR function, that can be encoded in DNA? So to do this, we turned to a basic logic function that had been done previously. And this is what's known as a NOT gate. And a NOT gate just does the opposite of whatever you tell it to do. And so if the input is on, the output is off. And if the input is off, the output is on. And so you can very easily build a NOT gate in DNA. And the way that you do it is the following. You just have a repressor. So this is a gene that produces a protein that then turns off a promoter. And so then we have an input that's a promoter. So you have RNA polymerase.
So you have a repressor that turns off a promoter, and now you input a promoter, and the repressor is going to shut it off so that it doesn't transmit the promoted output, and it will stay off. Very simple.
Flux that's going into the gate. It then produces the repressor protein, and this then turns off the output promoter. And so what you can see at the bottom of this slide is the response function. So as you turn on the input promoter, it turns off.
TMC, can you imagine that all this stuff is going on in our cellular and metabolic processes in our cells now? It's all going on. Nature designed it already. Isn't it just amazing? These guys are reverse engineering something that already operates in life now, and in life it's at much more vastly interactive and connected levels. But they're getting there.
Off the output promoter. So we can then convert this very easily into a NOR function just by having a second promoter upstream of this gate. So now we have two input promoters connected to each other in the DNA. And if either of those input promoters are on, then you have RNA polymerase flux producing the repressor protein, which then turns off the output. And so if you look at the logic function that's produced, you have the case where if both those promoters are off, then the output is on.
So let me just bring this to a real world example. Let's say we have normal cells, all right? A normal cell in a human or in any organism. And in a normal functioning cell, one or the other of these inputs might be on to cause the cell to produce a particular protein, maybe a structural protein to grow something. Well, let's say, hypothetically, that in a cancer cell, that when that cell, which is, they usually are, they're runaway cells that just multiply uncontrollably. Let's say that in that type of a cell, they've gone haywire and they've got both inputs going full bore. And now, what if you could set up a gate system in cells so that if they went full throttle multiplication, that they would get an inhibition? Well, guess what? Nature already does this. But what if we could enhance this in people whose natural function doesn't quite work as well as it should? We could build in, and this is a stretch, but I don't think it's outside the realm of being able to imagine it and even hypothesize the not so distant future of it, be able to build in better inhibitory mechanisms into cells so that if they start to go runaway, they either inhibit or they just get shut right off. That's a real world possible example of it. Cancer therapy. And we're talking electronics. We're talking logic gates, but it translates to cancer therapy.
But also be a good way to limit someone's life expectancy.
Yeah. Yep. And nature does that too. Nature kind of does that too already. I mean, you could turn it back on.
Yeah, you could program the body after your hormones drop after a certain amount of time, 50 years old, 60 years old or whatever. You get an average of, hormone baseline for 50 or 60-year-olds.
It could be done at many levels.
20. Yeah. And all of a sudden, all these different things just start turning on or turning off.
Right. So I mean, but that's what it's all about. Okay.
It's cool though. I got to get onto an airplane and I got to fly all over the country and I got to even vacation maybe over in Europe. I even got to see the Louvre.
Did you really?
I did. But you know what? my kidneys don't work anymore, and my eyes don't work anymore, my lungs don't work anymore, and
That can be fixed.
I've got these weird plaques in my brain that they attribute to Alzheimer's, but I don't really think it is, and
That can be fixed.
it can all be
All right.
fixed.
So right now, by the way, we're live, and if I were to take, I don't want to play. We'll get a real bad echo. I'll have to pause this in 1 second here. But if we were to take a look at what we were talking about, nature builds things. It's got inhibitory and promoting mechanisms in itself already, and it's not invalid by any means to let your imagination run with this. Designing molecules based on the construction of logic circuits, nanoparticles, tiny devices that know how to assemble themselves, and can be brought to specific locations in an organism or its cells. It's all in the design. Yep, it's all in the design. The genetic instructions, software, the power to create. This is where we are. And, we've created other things. I mean, we've smelted steel and we've built bridges and, we've made cars and computers and, wow, all kinds of things. We're very, very ingenious creatures. We are. And we're people are applying that ingeniousness now to this field. And it is the grail. This is like, if there was a final frontier, I got to say, that's probably it.
And it's only the case where if either is on or they're both on, that then you turn the gate off. And so that's this basic NOR function upon which you can build any other circuit function that you want. Now, what's really critical about this design is that both the inputs and the outputs of the gate are promoters. And so this means that you can take the output promoter of one gate and feed that as the input promoter to the next gate. And you can just do that in series until you've built up the circuit function that you want. But there's a problem with that. So if we go back to the Apollo 11 circuit boards, which are based on NOR gates, we found that all of these NOR gates are basically the same gate architecture, and they're physically separated from each other on the circuit board. And so you can use the same design over and over and over again and just connect them because they're physically separated. But a cell looks more like a burrito where all the biochemicals are pushed together and bumping into each other. And so if you had 5,600 NOR gates based on 5,600 repressor proteins, all of those proteins are bouncing off of each other and bouncing off everything else that's in the cell. And this can create interference between them.
And that's where that other term, TMC, what was it? That term that I mentioned a while ago in the show about the shape of things, the way that they're organized, the way that things are either...
Conformation?
Yeah, that's it. The conformation, the shape that things are in will either put certain active sites very proximal to each other so that they are likely to have the interaction that he's talking about, or they might be stretched or folded in another way that would make them so distant from each other that the likelihood of they just won't be, it's like, they're just too far away from each other to interact. So that's the next level of genius in life, which is the way it shapes itself at the molecular level. We see it with proteins, we see it with DNA, we see it with enzymes, such that either and this is where you were talking about whether something is expressed or not, that whether something is expressed or not might have something to do with the way that it's folded or conformed or shaped or not. So I'm curious what he has to say about this.
So one of the first things that we had to do was to find repressor proteins that wouldn't interfere with each other or with the cell. So to do this, we went into genomic databases, and using some computational programs, we found a number of different repressor proteins and then created synthetic promoters that responded to those repressors. And so what I'm showing as the grid is all of the different repressors and all of the promoters that have been designed for them and how they interact with each other. If it's a
it almost suggests that for every promoter that there's a repressor, because otherwise life would go haywire. Everything would be turning on, it'd all be firing all at once, and it'd be utter chaos. So that's where I think this leads to. In addition to shape, folding, and conformation, there's also the repressors out there.
red square, that means that they interact. And if it's blue, that means that they don't interact. So for example, in this case here, if you go to the TEDR protein, then it is binding to its promoter, but then isn't interfering with any of the other promoters in the system. And what you see then is for this set of repressors and promoters, there's a core set of about 16 that don't interfere with each other. And that means those are 16 gates that you can use together as part of a circuit design.
Wow. Don't you marvel at this? Just TMC, talk to me. Doesn't your imagination just run wild with the elegance of the way that we are built?
Yeah. What's also remarkable is the operating system that we all understand as Windows is now being put into the human species one script at a time.
Yep. Little by little. Yep.
Mm-hmm. Speaking of gates, and more gates.
What a name. Gates. And we're talking about gates. I got more stuff. We got we only got 11 minutes left. We didn't get to half of what we wanted to get to. So if all of us could be thinking about the possibilities to allow your mind to extrapolate to what we don't need to understand all of it, but just that you have a very complex organism, and it's got all sorts of these gates and promoters and inhibitors running and wiring between them. And it was really very simple when I was a kid and I had a 6-transistor radio it was a very simple little thing, and it worked. And 6 transistors. And now, if you were to hold an i9 Intel microprocessor in the palm of your hand, it's not 6 transistors. We're talking millions and millions and millions of transistors. But that's not my point. My point is, 40 years ago, 50 years ago, it's almost 60. Wow. 50 years ago, we built the 6-transistor radio. And now in the present, we built the thing that's got 1 million transistors on it. And it's so much smaller than that transistor radio was. So this literally guarantees you where this is going. It completely guarantees. You can have such total confidence in the ability for us to perfect the required material sciences necessary and to develop the manufacturing techniques and to interpret what is mountains of data of the existing code, okay, and then to decompile it, edit it, modify it, reassemble it, build it into a package. And as he said, you get your sequence that you designed on the computer, you send it out to a synthetic laboratory, which will sequence that exact string of digits that you want in nucleic acids, and they'll mail it back to you. And then you can use your best tools of the skill and the art to import that into a cell. And then it will do what you programmed on your machine. And that's where we're at right now. Well, that's where they were at when this video was done in 2016. Most assuredly, and we are definitely way beyond that now. So it's it's huge. It's absolutely huge. I'm going to let him go a little bit longer. There's not much else that we can do with our remaining 8 minutes of time, TMC, unless you got a brilliant idea, something that you want to really hit on. Figuring maybe, what do you think? Let him finish up.
Yeah, let's let him finish, and we'll see if we get to the.
He's done such a great job.
Yeah,
great job. I really like his style. He's very logical. Christopher Voigt, and this is Biology Techniques. You will find the link right below in our show notes.
So then for each one of these non-interfering repressors and their promoters, we then characterized the response function, and that shows how the gate turns off as the input promoter turns on. And that gives us the information that the computer can then use to figure out how to put together the gates to build a desired circuit. So now there were a lot of tricks that we had to use to get this to work.
Now, just keeping in mind, when he says build the desired circuit, you remember that diagram where he showed the computer and then the block diagram and then the string of A, T, C, and G, and then take that and basically email it to the lab that synthesizes DNA and then get the product back. So that's how we build the circuit, literally.
One of the big problems was that all of these different gates were interfering with each other. And so if you used a gate in one context, it wouldn't work when you tried to build it in another circuit. And so what we had to do is we had to create insulators that allowed these gates to be moved around in a lot of different combinations in order to build whatever circuit function somebody may want. And so to do this, we had to insulate all of the underlying parts. We had to make it so that you could interconnect.
So he's talking about circuits, but do you see the words that he's using right here? And it blanks it out when I put the mouse over it, but the word is ribosomes, which are cleaving messenger RNA, and which are also reading the code off messenger RNA. So we're using electrical engineering symbols and terms and techniques and machinery to work with ribosomes and messenger RNA. 6 years ago. What could we possibly be doing now? Wow. And you think we aren't? We are, or somebody is.
Motors easily, and you could stop the RNA polymerase flux from bleeding from one of the gates into the other, which would then screw up the computation. When we did this, we had to rebuild the gates to make a set of insulated gates that could all be used in a lot of different configurations. And this involved a lot of legwork in figuring out those parts that could make each one of these gates perform robustly without interfering with all of the other gates in the library. Then once we did that, we had
all the other gates in the library, and see, he's building a library, and those just get bigger and bigger, and they get more and more capable, more and more functional. There you go. This is how it's done.
This set of underlying gates that worked well, we then started to develop the software that would help us put them all together. So to do this, we turned to some software languages that had been developed previously in electrical engineering to help them build more sophisticated circuit functions. And so we turned specifically to a language known as Verilog, which has been around since the 1980s, and this is how electronic chips are designed. It's a hardware-independent language, meaning that you can generically describe a circuit function.
TMC, enter the new era of the profession of nanomolecular bioelectrical engineers.
Learn to code, bro.
Wow, man.
Just learn to code.
It is absolutely, we're moving. We are moving along. Okay, he's going. Here we go.
You can compile it to an Intel chip or an AMD chip or an FPGA or whatever you want. And what we did is we went in and hacked it so that instead of compiling a circuit to silicon, it would compile it to DNA. And this is the scheme for how it works. A user goes in, and this is a web-based program, and they can write the circuit function that they want using Verilog commands.
This diagram is courtesy of a guy by the name of Douglas Densmore from Boston University, by the way, according to the credit that you can't probably see at the bottom.
Then once they hit compile, the program then figures out the circuit diagram and the gates that are necessary in order to create that circuit function. It then goes into our library of insulated gates and figures out how to put all of these gates together in such a way that you can get a good circuit function. It then strings them together as a linear piece of DNA, and the output of the program is the DNA sequence, which these days can be sent off to a DNA synthesis company, and then a few weeks later you get your program. You can put it in cells.
Yeah, these days, that was these days in 2016. What about these days? That was those days. You know what I'm...
That's really, It's really profound what he just said right there. And he's just saying it all nonchalant. Yeah, you just mail out for your synthetic life form. And I'm being sarcastic. That's not what this guy's talking about at all. He's talking.
But it is. Yeah. I mean, he just needs to FTP it up or email it. You literally just electronically transfer the code for the life form that you want to build right out to the lab that makes it.
Yeah.
And test it out. Program is the DNA sequence, which these days can be sent off to a DNA synthesis company, and then a few weeks later you get your program. You can put it in cells and test it out. And so everything in the dashed box here is completely hidden from the user. We wanted to make a compiler where you just write your program and then out comes the DNA on which that program is encoded. And so this...
Right. Man. What world have we moved it? Write your program, and then out pops the DNA.
Yeah, I don't know about that.
Oh, wow. Oh, that time has come. Okay. I really don't want to stop today. All right. Can you sum this all up for us and bid our wonderful listeners and viewers a good week?
Yeah, sure.
My brain is fried.
Yeah. The biology piece is one of the driest pieces, but it is one of the most important pieces. That's what we are. It's what is not connected to the internet, allegedly. And that is what is being looked at to both create, modify, reform, reshape, mutate, remake, and ultimately master. And that is life. That's biology. We have to go through this. We're going to go through this. This stuff is critically important to understand the world in which we live in. Anyway, so onward until next week.